So I'm really happy to be hosting Drs. Tiziana Storlazzi and Dr. Roberta Carbone to discuss the recent PLOS ONE Lab Protocol article entitled "A Novel Method for the Isolation of Single Cells Mimicking Circulating Tumor Cells Adhered to Smart Biosurface Slides by Laser Capture Microdissection and the associated detailed protocol in protocols.io." Dr. Tiziana Storlazzi obtained her first degree in biological sciences from the University of Bari Aldo Moro in Italy. She obtained a specialization in applied genetics from University La Sapienza in Rome, followed by a PhD in genetics and molecular evolution from the University of Bari Aldo Moro in 2003. She was recruited as a researcher in genetics at the University of Bari Aldo Moro in 2004. Then, she became an associate professor of genetics at the Department of Bioscience, Biotechnology, and Environment at the University of Bari in 2016. She received a national scientific qualification as a full professor of genetics in 2018. She has focused all her research activity to genetic and genomic abnormalities with impact on tumor initiation and development. Particularly, her research interests focus on the identification of novel unconventional transcripts, including long non-coding RNAs and fusion and circular RNAs, which accompany genomic aberrations with a role in tumorigenesis. She has published more than 100 peer-reviewed manuscripts and accrued more than 3,500 citations. She's a frequent speaker at both national and international conferences and a holder of national funding for cancer research grants. Back in 2002, she was awarded an EMBO short-term fellowship as a visiting scientist at the Department of Clinical Genetics at Lund University Hospital in Sweden. In addition to being a researcher, she has over 20 years of teaching experience under her belt. Dr. Roberta Carbone is the Chief Scientific Officer of Tetis, a small Italian company based in Milan dedicated to the development of new technological solutions for in vitro diagnostics of solid tumors. She has gained extensive R&D experience in life sciences, driven by innovation with the final goal of introducing new diagnostic approaches to the clinic. She holds a degree in pharmacy and a PhD in forensic medicine, and obtained a master's in technology and innovation at the Bologna Business School in Italy. She spent several years at the National Institutes of Health in Bethesda, the US, and then 10 years at the European Institute of Oncology in Milan, Italy, focusing her research on technological developments in oncology. After joining Tetis in 2006, one of her significant goals has been to merge the biological world with nanotechnology, which allowed her to achieve the market introduction of new diagnostic device to analyze the genetics of hemato-oncological diseases. She is now managing the development of the diagnostic flow of a novel liquid biopsy technology to improve patient care and early detection of cancer by a comprehensive analysis of a simple blood draw. She has been responsible for several projects funded by the European Union and the Italian Ministry of Economic Development. She is a co-author of more than 30 peer-reviewed publications and has been the lead inventor in 5 patents. Tiziana, Roberta, thank you so much for being with us.
Thanks to you.
For pleasure. For me, it's a pleasure.
To kick off the conversation, can any of you please briefly explain what the protocol we will be discussing is about, how to use it, and why it is relevant?
Okay, so in our paper and in our protocol, we present a new method for isolating a single or a limited number of cells adhered to smart biosurface slides that we will call SBS slides herein and are patented by Tethys SPA. And these slides are glass slides which are coated with a nanostructured titanium dioxide. And these samples are used after a laser capture microdissection in order to isolate the cells. And these cells are then used for DNA extraction and subsequent whole genome amplification for further molecular analysis. So why have we talked about setting up this procedure? Because in recent years, the importance of isolating single cells from blood circulation is needed for several applications. For example, for non-invasive tumor diagnosis or for monitoring the minimal residual disease, or the analysis of circulating fetal cells for prenatal diagnosis. And also, there is the need for innovative methods. So, if we think about solid tumors, for example, a portion of cells is shed from the primary tumor into the blood vessel after a process that is called epithelial mesenchymal transition. Which results in the genesis of circulating tumor cells that we will call CTCs. And this cell can survive in the peripheral vascular system and initiate the development of metastases. Because of their essential role, CTCs are an instrument for diagnosis, for prognosis, and also as a predictive biomarkers in many types of cancer. So, for example, they can not only indicate minimal residual disease, but are also important as an instrument to investigate genetic heterogeneity that we know is the main cause of drug resistance. So, in this context, liquid biopsies and the identification and isolation of CTCs are crucial for non-invasive molecular analysis of cancer cells in patients. So, in our study, we use SBS slides to immobilize and characterize such cells based on previous results where breast cancer patients showed optimal results in immobilizing these cells. So what we did is to create mock CTCs by spiking cancer cell line cells in blood samples from healthy blood donors in order to mimic real CTCs. Then we prepare the slides with these cells and then transfer these cells to a laser capture station in order to isolate the cells, extract DNA, and perform a molecular analysis, in our particular case, to detect mutations. Okay, so this protocol is suited to use single cells for mutation detection.
Thank you so much. And to understand the significance of the technological development, and I know that you briefly mentioned this in the paper, but can you elaborate what was the state of the art when you decided to develop this technology? Was there any other way for scientists to detect a very small amount of circulating tumor cells? And if so, why those protocols didn't work for you? Why do you think it was important to optimize a new protocol?
Okay, so for this question, I can say that there are to date several technologies that are already being developed for isolating and characterizing CTCs from blood, even at the level of single cells. Those methods essentially are usually based on the enrichment of CTCs or depletion of white blood cells using specific antibodies, filtration by size or deformability, separation by macrofluidic systems. For this system allows to collect, let's say, a part of the CTC. So there is a selection before the collection of the CTC. SBS slides allows the collection, immobilization, and analysis of the all white blood cell fraction and also the rare cells that are contained, that can be there. Minimizing sample manipulation. So we can have a fresh blood sample processed by an automated sample preparator that seed the cell suspension on a slide and providing a monolayer of white blood cells on the surface. And then this single cell can be further characterized with different kinds of analytical protocol. After the immobilization of the white blood cells and the fixation of the cells, there are different kinds of analytical protocols that can be applied, like immunofluorescence, FISH, bright field, and therefore also single cell analysis by picking of a single cell and then going up to molecular analysis. Previously published protocol mainly relied on a different method for single cell isolation from commercial slides, so different slides compared to our slides. And this method is called cell picking. The cell picking uses a capillary made by glass, as a needle can aspirate the cell from the slide. So this is a very delicate procedure, which has a limitation since, for instance, it cannot provide a precise way to detach a single cell, especially in the context of CTC clusters. So many times in the blood of patients, especially in metastatic settings, can be found these very important biomarkers of the aggressiveness of the disease that are the CTC clusters. CTC clusters are cells of CTCs that are bound to other cells, even with white blood cells. And it's very important to be able to isolate one by one. And this is not possible with a not precise method like cell picking. And so this is one reason why we didn't want to use cell picking, especially in this context. Then also there is no clear literature showing that the possibility to reach 100% of efficiency of cell detachment from the slide using cell picking, as we did with laser capture microdissection. So in addition, other methods start for the isolation of living cells in suspension. And these are living cells compared to our cells that are fixed cells. And for the molecular analysis, they use a different molecular protocol for the genetic characterization. So in summary, we had to develop a dedicated protocol for two main reasons. The first, we wanted to use laser capture microdissection to precisely and efficiently isolate single cells. And this was possible thanks to the specific nanocoating of SBS slides, because the nanoparticle that can allow the detachment upon microdissection of the single cell from the slide. And then secondly, we had to develop a dedicated molecular protocol capable of efficiently working specifically on fixed cells. So these are the two main reasons for the dedicated protocol and new protocol for single cell molecular analysis.
And I would like us now to go a little bit deeper into your protocol. So if you can tell the audience, what are the most critical steps?
Yes. So first of all, we needed to set up the laser parameters in our instrument, which is Leica Microsystems LMD6500 platform, which is optimized to microdissect cells from specific LMD membrane fold slides. So we needed to change, of course, and to isolate cells from SBS slides and limit the possible negative impact of the laser beam on cell nuclei, we just set up appropriate laser parameters that were, for example, power, aperture, speed, and specimen balance. So we made several attempts, and finally we came to the correct combination of these parameters that are reported in the paper, but just to mention, for example, is power 32, speed 22, etc. Secondly, another problem was to optimize the recovery rate of the cell microdissection. So even in this case, we needed some time to optimize, but finally we reached almost 100% recovery rate of the cut nuclei. Thirdly, what we needed to do is to find out the best performing kit for whole genome amplification, because there are several commercially available kits, and we tested most of them. But finally, we came to the AmplyOne as the most efficient in the amplification, but not only in the amplification process, but also in obtaining high-quality DNA for subsequent molecular analysis. So this was also a very important step. Finally, we needed to be sure not to introduce contamination by the operator. So there are two main steps in which an operator can introduce contamination. The first one is during the microdissection, because it's a very critical step. Because for our instrument, for example, the cell falls into the recovery tube by gravity. So of course, the operator must be very careful not to introduce contamination. Okay, and the second step is also during the whole genome amplification process. So what we did is to profile the DNA by STR analysis. So we tested the operator DNA and the cut nuclei DNA, either after all the steps, after the cutting and after the amplification. And we could exclude the presence of any contamination by the operator. So these were the 4 main critical steps of the procedure.
And while you were describing these 4 steps, you mentioned a little bit the need for troubleshooting, setting the right parameters and for the laser. And also you mentioned that you tried several genome amplification kits and decided for one specific one. I hope that you can elaborate a little bit more on what else you tried that didn't work. You mentioned the laser, you mentioned several genome amplification kits. Were there other things that you tried and didn't work and didn't make it to the final version of the protocol?
No, because these were the 4 main problems that we encountered. Also because the procedure is very easy. And after the identification of CTCs by immunofluorescence, and this is very easily performed by identifying cells that are negative for hematopoietic marker CD45 and positive for pan-cytokeratins markers, then the coordinates, the slide coordinates, are transferred on the laser capture platform. So it's very easy to identify the cell that then is counterstained by GIMSA, and then we can cut and then all the mixture for the PCR is present in the tube, and then the DNA is, of course, used for subsequent molecular analysis. So we didn't have any problem. What is important is to be pretty sure that the DNA is void of the contamination by the operator. So this is very important for all the procedure.
And at the beginning of this conversation, you mentioned that the way that the protocol was optimized and developed was by taking blood from healthy patients and spike it with samples of patients with pancreas and colon carcinoma cancer. So my next question would be, have you tried the protocol in other types of cancer? And second, have you applied the protocol to samples from real cancer patients?
Okay, so the answer is no. There is a reason for that, because we are searching together, myself and Roberta, for additional fundings for this research. Is a tricky step, and also because to work with patients on this project, of course, you need ethical permissions. So we are waiting, of course, for ethical permissions, but most of all dedicated fundings, because this is an expensive research. You have to consider that whole genome amplification kits cost a lot of money. So so it's important that this research is supported by dedicated funding. So this is the reason why so far we haven't tried with other tumor types, and mostly on patients.
If I can add, sorry, if I can add, let's say a comment on this. To optimize this protocol is very important, because it's very important to validate the fact that a single cell is a CTC. So should be very, stressed, because when you find a cell with a biomarker, like for fluorescence, you find a biomarker, a positive for epithelial marker, negative for hematopoietic marker, a positive for a tumor marker like estrogen receptor or HER2 in case of breast cancer, or other biomarkers. It's always a question mark if that cell is real a CTC. So this is a very universal problem for all the people working with biopsy and CTC. Therefore, the verification, the evaluation in molecular analysis, a single cell for copy number variation is of paramount importance to confirm that that cell is a CTC. Then also having a specific molecular detail mutation is even better, because it can provide information regarding targeted therapy. So the molecular analysis of a single cell is of paramount importance for the characterization either of the sample itself and the patient. So this is the importance to continue the studies in single cell analysis and CTC.
Thank you so much, Roberta. I would like us now to change gears a little bit, and I hope that you can tell us a little bit, this is a beautiful protocol. So you develop it, you optimize it, you share it in the platform, but you went one step further and decided to have it peer-reviewed. Can you tell us about the motivations to publish the protocol as a standalone methodological paper?
Well, as a single cell isolation is so important for multiple applications, we believed that it could be crucial to share this protocol with the scientific community in order to spread this piece of information with the aim of finding it usable in other laboratories that could provide further input to optimize it. So this was our decision, and I think that it is very important in science to share with other people so that they could also improve the protocol itself.
And can you tell us how the feedback that you receive from the peer reviewers at PLOS ONE help you shape the final version of the protocol?
Yeah, so the protocol itself was not modified according to a reviewer's suggestion, but what was very important for the input received by the reviewers was in improving the clarity of the manuscript. For example, they helped us very much in clarifying some points in the results. For example, in describing better the laser setting procedure, or in the discussion conclusion, for example, clarifying the fixation procedure or the maintenance. They asked if we were able to maintain the cell heterogeneity on SPX slides, and this is the case, or also discuss about the safety of the laser on the microdissected cell DNA. So this was very important for us to clarify some aspects of the methodology that maybe was not so clear from the beginning.
And I know that, this is actually my last question, and I know it can be a little bit tricky to answer because this is a very recent protocol. It has like just over a month that it was published as a paper. But if you can speculate, or if you already have data or anecdotes suggesting, what has been the significance of publishing this paper for the career of both of you?
Okay, so I work in a university. I work in the University of Bari, and for me, it was not only a pleasure but also an important step to perform a synergistic work with a small medium enterprise company such as Tethys SPA. So, and also I have to say that this work is the result of a work that was developed during the PhD research activity that was funded by the Italian Ministry of the University and Research. So this lab protocol, this paper, indicates in my career the first goal concerning the interaction between research activity in my lab and that developed in a company, so in Tethys. It was a pleasure to work with Roberta, I have to say.
Thanks, Tiziana. Okay, so from my side, I have to say that for Tethys, actually, and also for me, this possibility represents a result to the paper as a result of a continuous effort that we have in the company to foster innovation through the collaboration of public and private institutions. Because actually we have the final goal to make such innovation available to the community, and in particular advancing the diagnostics for complex disease like cancer. And we do believe that with innovation, if not now, but maybe in 10 years, we'll be able to manage better the disease, a very, very terrible disease like cancer. So even if this protocol seems a small part of the overall picture, of overall scenario, we believe that it's a very important piece of the puzzle. And then I'm very happy to have the possibility to collaborate with Tiziana, with the university, with the colleague of Tiziana, and publish this paper in PLOS ONE.
And so before we wrap up, is there anything that we haven't touched in this conversation that you would like to mention?
I just would like to mention that also to publish a protocol that is with the format of the protocol helped us very much in describing better the procedure, more than maybe as publishing just a paper, because it was really helpful to describe the procedure step by step and define the details of each method passage, including how the method, for example, could be changed for other application. As I said at the beginning, for example, this procedure can be also used for the isolation of fetal cells, for example, in the blood. So, or maybe for other tumor types. So, to us, it was really good to have the procedure better described for other laboratories, of course.
If I can add something on this, I completely agree with you, Tiziana. Actually, since we are in a company and we are dedicated to develop standard operating procedure according to the ISO, to the standards for developing in vitro diagnostics, writing a protocol and a lab protocol, even as an article, it's very useful to spread the education, to write correctly a protocol to be able to, for other people to replicate. And then following certain regulations that are more, let's say, used in a company, because you have to follow certain regulation to be able to develop in diagnostics. But again, it's important to share this knowledge even with the basic research to be able to have exact protocol that can be replicated in other lab. So I completely agree with you.
Well, Tiziana, Roberta, thank you so much for your time. Thank you so much for your insights and for helping putting the significance of this really nice protocol in perspective.
Thanks to you, Gabriel.
Thank you very much, Gabriel.
Thanks.